one of the reasons was the opportunity uh, where I saw that I can contribute with something new to where I live and stay. So this is the first of its kind company in India, which is focused on ribosome and mRNA translation. Pretty proud to say that and present TranslaTale in that light.
Gnaneshwar, yeah, welcome to this session that we are having. This is a mentorship session that we've been having for many years now. And Virender Singh, who's here, heads Biopatrika, who in the forum in which we do this through this session. So we've interviewed probably like 70 odd people over the last 5 years. And welcome to this session. So I'll quickly introduce you, and then maybe I'll hand it over to you, and you can do a presentation. So Shirish and Tripti, who are here, Gnaneshwar is a scientist, has done a lot of work in translational medicine, not translation. It's both translation and in terms of translation medicine, as well as understanding the translation of proteins and RNA. So we'll talk about that. I'll ask him to give a brief introduction of his experiences, but I've been working with him and his company TranslaTale for last couple of years, and he's doing some very exciting work in Bangalore. So over to you, Gnaneshwar. Thank you for coming to the session.
Thanks a lot for having me over, Naren. Hello, guys. It's nice to be here with you guys. So, yeah, hi, I'm Gnaneshwar. I'm the CEO and founder of TranslaTale Biosystems. We are a drug discovery company based out of Bangalore. We are a very young startup founded in 2022. And right now we are into 1.5 years of our incorporation and operations, going successfully so far. And I'm really glad to share my journey and my experience here with you guys. I'll start with where it all began for me. By background, I'm a science graduate. I have a bachelor's in fisheries science, which is a very less heard of subject. It's a agriculture or veterinary allied sciences subject, where we study about the marine biology resources, the economy that runs, that is driven by marine biology, where it gives a lot of livelihood to different people. We study many aspects ranging from the biology of the marine systems to economics, statistics. Even we had to learn swimming. We even had a scuba diving course too. So it was pretty exciting to study here. This campus was part of Agriculture University, the GKVK, as you would know, in Bangalore, the Gandhi Krishi Vignana Kendra, as it is popularly known. It had its Mangalore campus offering fisheries science courses. It still does exist. It's one of the first colleges in the whole of Asia that started offering this curriculum way back in 1960s. And yeah, I mean, we are very proud to be part of this college because it offered a lot of dimensions, not just in terms of science, but also in terms of extracurriculars. It helped me explore a lot. Although this was a professional science institute, it offered me to explore much beyond the biology courses that it was presenting. And at that time, I got very much attracted towards the booming space of biotech. And obviously, on the side, I had to drive my passion too. So I was obsessed with basketball, and I got to represent my university several times while I was studying here. That was my way out of the rut from studies and science. So both biotech and basketball became very dear to me at this place. And I sort of knew somehow that I have to continue in the biotech space. And to do that, the next best option I had was to do my specialization through master's in microbiology, which happened at the same place, uh, being the first of its kind college in Asia. It had a very good faculty and department lab for studying microbiology, which helped me continue my passion and obsession with biotech in this same campus, where I could work with two very well-known faculty at that time, both husband and wife, Indrani Karunasagar, Dr. Indrani Karunasagar and Dr. Idya Karunasagar. So this helped me develop my concepts in immunology, biotech, microbiology, and also human diseases. Because fish would be consumed by humans, there was a lot to study through my master's in fisheries microbiology about human diseases and not just fish diseases too. So it gave a very good and solid foundation for me in areas such as immunology, biotech, microbiology, food technology, and all these areas too. So after my master's in fisheries microbiology, I had found my way out of this campus to do my PhD again, uh, where I had an opportunity to pursue what has always been a constant in my career, the fish, where I got an opportunity to start using zebrafish as an animal model to study human diseases, where while I was doing my PhD in Japan at University of Miyazaki, where I was working with Dr. Kenmochi, who was leading the group to understand how human diseases are caused due to defects in ribosome and mRNA translation. And it has stuck with me for life now, this obsession and passion to work on ribosomes and mRNA translation. It is so exciting to be in this space. These two blobs that you see actually represent the ribosome, on which the messenger RNA translates into protein. This is the hub where all the genetic information has to translate into protein. And it was really wonderful to learn how diseases are caused as defects in this complex where the messenger RNA is translating, and especially understand it by using zebrafish as an animal model. And this is where I got an opportunity to get my PhD in creating by creating disease models using zebrafish to study human diseases. That basically led me to explore more of ribosome and mRNA translation for my entire life till now. And yeah, through my work and efforts, I was also recognized as a valedictorian for my PhD graduation ceremony, which was a very wonderful opportunity to represent the entire batch of PhD students to give the valedictorian speech. So I kept on developing this passion and obsession to work on what has been constant throughout my career uh, till this point, which is working on fish and explore its relevances to human diseases, right? So I continued with this passion by moving back to India in 2014, where I took up postdoctoral fellowship in neuroscience by working with Dr. Vatsala Thirumalai at the National Center for Biological Sciences, which is part of TIFR Mumbai. This is in Bangalore. And here, although Vatsala's lab is a neuroscience lab, hardcore neuroscience lab, and she had nothing to do with ribosome and mRNA translation at that point of time, she encouraged me to explore what has always been my passion. And I was looking for labs where I could bring something new to the table. And this was a lab I felt where I could bring my expertise in molecular biology, in molecular uh, biology of ribosome and mRNA translation, and develop something new in the lab, where I got to continue my science and obsession of working on ribosome and mRNA translation. And also simultaneously working on these aspects in zebrafish model, which is which is one of the great alternate preclinical models currently available to study human diseases. So after my postdoc fellowship, I continued my obsession uh, with ribosomes and mRNA translation. Although you can see at various stages in my career, I've been making shifts from fisheries science to microbiology to studying human diseases. I've been maintaining one or two constants, which always have helped my bearings to be right and heading in the right direction. And with this in mind and my ambition to have a company that is focused on ribosome and mRNA translation to to discover therapeutics, to discover diagnostics, I founded TranslaTale Biosystems with my co-founder, Dr. Saurabh Mahajan, who was also my former colleague at NCBS TIFR. We both founded TranslaTale Biosystems, which focuses on ribosome and mRNA translation to discover targets, to discover biomarkers, to discover drugs that are very relevant to treat human diseases. And here again, we maintain our interest in using zebrafish as our preclinical animal model. And we were recently awarded the BIRAC BIG to to take our plans of developing our science at TranslaTale forward. Currently, we are incubated at CCAMP, which stands for Center for Cellular and Molecular Platforms, which is a deep science and innovation center, basically an incubation and accelerator, which is based out of the same campus as NCBS in Bangalore. And we continue to take our science forward to benefit the society in some way or the other by translating our science from bench to bedside innovations at TranslaTale Biosystems. And this is what TranslaTale is focused on, where rest of the space in biotech and pharma is heavily focused on DNA, mRNA, and proteins. We at TranslaTale are focused on this checkpoint in information flow of this pathway from genetic code, which leads to proteins. So ribosomes sit exactly at this, crux in this pathway where they translate the messenger RNA into proteins. And we see that they are relevant in a lot of diseases, ranging from cancers to neurological diseases and even several rare diseases. So at TranslaTale, we are focused on bringing solutions to pharma, to the biotech industry, with our focus and obsession with ribosomes and mRNA translation, and bring ultimately novel class of targets for which we can find first-in-class drugs. So this is what we are primarily focused on helping the society with through TranslaTale and our efforts, basically to bring in platforms that enable drug discovery and bring in molecules through our efforts that can impact lives, human lives, and treat diseases. So this is how my journey has been. And this is my co-founder, by the way, Dr. Saurabh, about whom I was talking about, with whom I co-founded TranslaTale Biosystems. These are a few aspects of my journey, and with this, I will probably be open to discuss further details of what we do and what the rest of our journey has been like.
Great. Thank you, Gnaneshwar. And I think first let's open it up for a few questions. You can unshare your slides, and
Sure.
maybe what you can do is I don't know if you have slides or you want to talk about it. How do you look for tissue-specific translatomes using the zebrafish model, and what is the advantage of the zebrafish model over, let's say, a mouse model in discovery?
Yeah. So zebrafish.
Nice. Yeah, sure.
Yeah.
Go ahead.
Yeah, thanks, Virendra. So zebrafish has to basically study the fundamental biology of diseases and also to discover drugs, mainly because of its quicker development, which you can monitor under the microscope. It's pretty transparent in terms of its internal organ systems that you can see through a simple microscope that is basically found in any of the labs, where you can literally study the organ development, be it brain, be it eye, be it heart. You can even monitor the heartbeat, the rate of the heartbeat, the blood flow. All of the internal organs are visible through its transparent skin at earlier stages of the development. So that is the beauty of using zebrafish as an animal model, where you get to see tissue-specific development in finer detail. So that helps both fundamental biology people who are after understanding the development of the tissues or its development in a disease or its development in response to drugs. So all of this is possible by using zebrafish as compared to other animal models such as mice or rat.
Maybe some other questions. I have a few more, but yeah, Shirish.
So thanks. Okay. Someone
Oh,
else.
sorry.
Maybe
No, it's okay. It's okay. Virendra, go ahead. Go ahead, please.
Yeah. Thanks a lot for sharing your journey, and it's good to see that you stuck to one thing and got expertise in that field, and now you are translating it to something which might be useful in future. So I have a question that like your platform is to screen molecules that could be then like transferred to some other company, or how you see your like pipeline could go ahead in this?
Yeah. Thanks for that question. So regarding the first part of the question, are we, using platforms to help companies discover their molecules? Yes. So that is one aspect of our business development strategy, where we want to help other pharma companies, established pharma companies, both in India and abroad, to develop their molecules that can specifically target ribosome and mRNA translation. That is an up and emerging trend in many established pharma, where they're trying to partner with smaller startups who can offer novel platforms where they can screen their molecules. So that is how we are looking at as one of the options. But we would also be interested in having our own molecules at some point sooner or later in the pipeline.
Okay, nice. But do you think like it can be made into a like high throughput platform? Like if you
Yes.
consider C. elegans or
Right.
like Drosophila larvae, those could be
Uh-huh.
put into 96 wells, and you can,
Right.
based on like fluorescent, you can make a readout. But
Right.
could this be translated to like high throughput?
Yes. So zebrafish also offers that advantage. Just like you can put Drosophila in several well formats, right? And do high throughput screening for hundreds of drugs in a day. Same assay, same high throughput format is also possible with zebrafish larvae, which have quick development, and they are very small in size, so they can fit into these tiny wells in the high throughput format, where you can test all the molecules, where otherwise you would be screening on cells grown outside the body of the animal or the human patient. You would be directly testing it on an animal on thousands of in the high throughput format.
Nice, thanks.
Shirish, you had the question, right?
Yes. So I have actually three questions. So the first one is, so since you start doing the experiment on zebrafish till you see the outcome of, let's say, a particular molecule, how much time does it take? Okay. Second is, see, I mean, since I worked with the translation East system, I find that was, we can also use that as a proxy for eukaryotic systems, right? Even for higher eukaryotic systems. So in your experiments, do you like do your initial experiments with East, since it might give you a quicker result compared to, say, zebrafish, and then take those experiments forward to be repeated or to be tried on zebrafish? That is the second question. And third is, see, zebrafish is not a mammalian model, right? Your final goal is to have this to be in society for human purposes. So in that case, do you find that switching to a mammalian model might give you a better understanding of the outcome that you might see in, say, for human cases?
Got it. Thanks, Shirish, for that question. So did you say translation as an mRNA translation
Yes,
in
translation
East?
control.
Okay,
Yeah.
great. That's very good to know. Happy to meet a fellow mRNA translator. So yeah, Yeast has some benefits to enable screening drugs as well for for targets that have high resemblance to human or the mammalian ribosome and mRNA translation factors too, right? But also what happens with yeast is there are gaps where you don't see that resemblance with the human system in many places as well. But zebrafish gets much closer to human ribosomes and mRNA translation factors, where in our observation we see 80 to 85% similarity between human and zebrafish ribosomes. So that is one advantage of working with zebrafish system. I'm not saying East is still a bad system. It is still good where there is a lot of similarity, where you can confidently go ahead and use it. That I think helps answer the first part of your question, right?
Yeah.
Yeah. So can you please repeat what was the second part of your
Yeah.
question?
So
To be more
then
clear.
how much time do you, I mean, since you start the experiment till you see any outcome in zebrafish, how much time does it take? Because in East, I think it can be around 2 to 3 days maximum, right? Even if you're talking about repression or something like that. So,
Yeah. So in terms of turnaround time, it's more or less similar to East. See,
okay.
zebrafish takes 24 hours for its you know uh, embryonic development. As soon as
Okay.
it starts fertilizing at the zeroth hour, it
Mhm.
it in 24 hours you can see its eye, you can see its brain, where you can see the forebrain, midbrain, hindbrain system clearly. At some point in the session I would run that video. I'm not able to find it in my drive. I thought I had it, but so what I'm saying is the advantage with zebrafish is the fast development, where under 24 hours you will see vital organ development, eye, brain, heart with a heart beating. You can see few red blood cells in circulation. Musculature is very clearly visible. So some of these major organs development is pretty clear. That helps you understand what is the effect of a disease within 24 hours, right? And if you uh, let it pass 2 more days, you will see much of the other organs also start developing, which helps you understand its effect on multiple organs in a stretch of 2 to 3 days. Within a week, most of the organs. So this gives a holistic, wholesome view of what is the effect of a disease factor, a target, when you manipulate it, or when you treat it with a drug, right? So this is the main advantage of zebrafish. It definitely cannot translate everything directly, and help you apply it to humans. It is one model system that can bridge the gap between in vitro and other in vivo systems like rat mice, where the discovery process is very late, right? It lasts for 1 to 2 years with rat and mice. And with in vitro, you only get few data points mapped, which are very critical, of course, to move your understanding forward. But there is a huge gap between these two systems that zebrafish can very well uh, cover and give you quick pointers with a comprehensive readout in under a month. So that's one powerful advantage with zebrafish. And also, I think Virender also asked, where does it position when you see this against C. elegans or Drosophila, right? So Drosophila and C. elegans are non-vertebrate models. Zebrafish is a vertebrate, and there it it helps the gap between an in vitro and in vivo system be filled with uh, with a vertebrate animal model.
Well, Gnaneshwar, I think I guess I'll ask the question from an entrepreneurial perspective. Maybe you can talk a little bit about what it has been for you to, transition from academia to industry. I mean, to entrepreneurship, because you had an option of going and working in a company or something like that, right? Why entrepreneurship?
Yeah, that's that's a very tough one to answer, actually, although I'm an entrepreneur now myself. But one of the reasons was the opportunity, uh, where I saw that I can contribute with something new uh, to where I live and stay. So this is the first of its kind company in India, which is focused on ribosome and mRNA translation. Pretty proud to say that and present TranslaTale in that light, uh, because we really are the first one in this space, which is the opportunity that I was talking about. But also this opportunity presents with a lot of risks and challenges. Being the first of its kind is not easy to convince the space that we are relevant. We have to create our own niche in this space, where we have to find our own kind of customers, our own kind of business, our own kind of even raw material providers. So there are multiple challenges that I had thought of, which actually made me not think of pursuing entrepreneurship at one point of time. But then it's an experience that I was waiting to to also have in my life, because if not with what I have right now, which has been literally my lifetime's experience of working on ribosome and mRNA translation.
Your journey has been quite interesting. From transitioning from academia, applying for the BIRAC grant, and getting funded. I think that was a good exercise for you. I from what I've seen you is it's sort of intentional, but there's also opportunistic, because you applied for the grant and you got funding, and so now you're here.
Yes, that's true. Many things fell in place at the right time. And thanks to your mentorship, Naren, which actually, opened many doors to understand how entrepreneurship journey should be pursued. Because otherwise, without mentorship, entrepreneurship journey is impossible. So thanks to you for your mentorship there. That in a way paved way for a lot of confidence. Otherwise, most of the entrepreneurs, although they display a lot of confidence in what they're doing, there's always this constant thrust behind us that what if this fails? What if this doesn't work? And the way forward is probably having a proper mentorship. And I think there we were lucky to have mentors like you, Rams and Vatsala, who helped us see the road ahead to pursue that as well. So, in that sense, many things fell in place. One was mentorship, which was key to actually apply our knowledge in the form of a proposal for a grant that could fetch us the money we need to run our program, run our idea, and get us to a proof of concept stage. So that is what I meant by many things also falling in place, and also CCAMP providing us the incubation space and support in this journey. That also matters. So many things fell in place there for us.
Okay. Unless are there any questions from Nagesh, Shirish, or Trupti? Otherwise, I'm going to do one exercise.
Yeah. I just have one question. Okay, actually.
Yeah.
Gnaneshwar, I mean, I've been curious about this, right? When somebody starts a, let's say an entrepreneurship venture, and that too in a field of biotech, which is in itself has its own risks, right? So how
Right.
can we be sure that the big picture that we are imagining or we are seeing is the big picture, that is how it's going to turn out? I mean, how hard is it to convince somebody else, especially the your fund or officers, to convince them that okay, this is the correct picture, this is how the future is going to turn out, and this is a essential research that we are into. So have you find it difficult? I mean, obviously you would have, but how you have conquered that stage?
Mm, yeah, that's a very brilliant question. That makes me very nostalgic about how it all started. So that is where I was saying it's very important to talk to mentors. Mentorship matters a lot in entrepreneurship journey. I think many people talk about it, but it is one of the things that helped us, where we, of course, appointers like Naren, Rams, and Vatsala with our idea. They asked us some really tough questions, which obviously later on reviewers, uh, grant agencies, investors, right? All of them ask because they have seen something ahead of us, and that experience matters. So getting that on our side, getting our mentors' interest in us on our side was very critical, and that helped us to understand the whole space. Otherwise, me coming from academia, trying to set my foot in very cutthroat space in business, especially pharma and biotech space. It was pretty scary. We weren't much worried about competition per se, but definitely we were worried about securing a grant, convincing our investors, our grantees. So there, our mentors helped us figure out the nuts and bolts of the process, where what needs to go in beyond just science. Science is one bit of it, one important bit of it, science and technology, because we are a deep tech, deep science company. But where exactly are we going to, take our science forward? What are the nuts and bolts that are very critical uh, to build the architecture of the company, right? Be it our platforms, be it our business strategy, be it our uh, later on projections to monetize once we are successful in building our platform or drugs. All of it, we had our sessions, therapy sessions, and very powerful, one-on-one sessions with our mentors. So I would credit them, and also, it's very important to talk to many people, as many people as possible. And this is something that Naren constantly keeps asking us to do even now, which we are still doing, to keep figuring out the mindset, the psychology of people in the system, from where we can derive, our plans, so that we can fit our science in the grand scheme of things that people have already in place. So talking to people, different people, it could be even grant agencies. Why do you fund a project? We could be asking them this question. Why do you not fund a project? Although it was interesting, right? We could ask them these questions. So talking to incubators and accelerators also gave us an idea what to do, what not to do. And a lot of self-help books too. Reading is very important. Reading a lot of blogs, investment blogs from venture capitals, right? From Y Combinator, the prime, uh, venture capital that is, um, very, very much sought by many startups. So we had to read a lot, talk to a lot of people, scientists, incubators, accelerators, customers, investors, everybody literally, to understand the whole game.
Right.
Great. So
Thank you.
thank you, Gnaneshwar. So what I'm going to do is, in the last section of what we do here is I do a rapid fire question. So you have to answer
Okay.
questions quickly. I'm going to ask you like 7 or 8
Okay.
questions, and then you have to give them without too much thinking and short answers.
Okay.
Ready?
Okay.
Okay, here. Describe yourself in 3 words.
Yeah, passionate, very obsessed with certain things, and like to uh, bust my stress very frequently.
Okay. Your favorite food?
Um, I don't have favorite foods.
Okay,
I think
that's
it
okay.
would be, yeah.
You can pass. Your
Yeah.
favorite teacher in school and why?
In school. Did you say school, Naren? School and college?
Yeah. School.
School. Okay. Yeah. Mumtaz ma'am.
And why?
She stood out in the way she cared for her class. Never one got punished, but everybody was disciplined. Everybody were well behaved, and we
Okay.
had no qualms in the way she handled us.
Okay.
She was very caring, motherly.
Hmm.
At Saraswati Vidya Mandir, I have to quote that.
Hmm.
She was our class teacher from 6th to 7th
Wow.
grades.
Wow. Your maybe 2 or 3 people who inspire you
Hmm.
or role models.
Yeah. Oh, Venki Ramakrishnan, of course.
Hmm.
I can't not mention him for his contribution to ribosome and mRNA translation and him winning Nobel Prize. The contribution has been immense. So one is him. The other would be you said 3 people.
Any amount, even one is good,
Okay,
but
okay.
if you can think of anybody else immediately, that's why these are rapid fire.
Yeah. So to drive my passion, I think Dr. Shyam Sundar, who was our food technology professor at our college, was very instrumental. His foundations in food tech was solid. His preparations in food tech were quite amazing. Although he wouldn't taste it, he would offer other people because he was a vegetarian, and fisheries science had a lot of fish processing technologies. He wouldn't consume it, but he had excellent hands in food technology labs. Yeah.
What are the most impactful events in the world in your mind that are going on?
Impactful. Yeah, the space tech, as catching bus to space. That's pretty impactful. The other is our shift to green energy. I think that is also going to be very impactful.
Very good. What superpower would you like to have?
Yeah, superpower enough to turn pollution out of our ecosystem. That would be amazing to have, I think.
And then maybe last two, maybe. Would you rather know every language in the world or be able to talk to animals?
I think be able to talk to animals would be interesting.
Okay, good. And then I guess, the final question is any final words of advice or anything that you want to share with, mentors or who are mentees who are looking to transition either from academia to industry or trying to think about entrepreneurship? What are the kind of things, any advice that you could end with?
I mean, talking to as many people, as you keep saying, Naren, that would be the best advice. Talking to people to figure out the process quickly. That helps you position yourself ahead. You don't have to waste time to start doing that, or be shy not doing that. Just talk casually. Most people are willing to help. Most people are willing to share their journey because they obviously feel like there is value in what they have experienced and what they have achieved. So they will be more than happy to share it with you and help you position yourself better.
Great. So thank you so much. Thank you for coming today, sharing your experiences. This was a very insightful thought of from where you started in fisheries and continue to work on zebra fish, of course, completely different stories, and your journey in between. Yeah. And thank you for everyone for calling in. And see you next month.
Thanks a lot, Naren.